Hello and welcome. Today we're going to dive into the incredible microscopic dance that makes you, well, you. We're talking about the process that makes every single one of us a one-of-a-kind genetic masterpiece. It's called meiosis.
So think about it. Why are you genetically unique? Why do you have your mom's eyes, but your dad's sense of humor? Why don't you and your siblings, unless you're identical twins of course, look exactly alike? Well, this amazing variety isn't just an accident. It's the result of a really precise and elegant cellular process, meiosis.
So here's our game plan. First, we'll get into why meiosis is so important. Then, we'll walk through its two big acts, Meiosis I and Meiosis II. And finally, we'll put it head-to-head with its cellular cousin, mitosis, to see how they stack up. Sound good? Let's do it.
Okay, first things first. Let's start with the big question, the so what. Before we get into all the nitty-gritty of how it works, we really need to understand why it happens. What's the whole point of meiosis?
This quote just nails it. The raison d'être, the entire reason for being, is genetic shuffling. Meiosis is nature's way of constantly shuffling the genetic deck. And all that shuffling creates variation, which is the raw material that lets species evolve, adapt, and survive.
So, what does meiosis actually make? It produces these really specialized cells called gametes. You know them as sperm and eggs. And the key word here is haploid. It just means that these cells have only half the normal number of chromosomes, just one set, instead of the two sets that are in pretty much all of our other body cells. And here's why that's so incredibly important. You see, meiosis is what we call a reductional division. It starts with one parent cell that's 2N, meaning it has two sets of chromosomes. It goes through its 2 rounds of division and out pop 4 N gametes, each with just a single set. This is absolutely critical because it ensures that when a sperm and egg eventually get together, the new organism has the correct 2N number. It's perfect biological math.
All right, let's get into the mechanics. The first and honestly the most complex part of this whole thing is Meiosis I. This is where the real magic happens. This is the great genetic shuffle that creates brand new combinations of genes that have never existed before.
The main event of Meiosis I is separating what are called homologous chromosomes. The easiest way to think about these is as pairs of instruction manuals. You got one from your mom and one from your dad. Meiosis I is the process that doesn't just decide which manual gets passed on, but actually swaps pages between them first.
Here's a look at the whole journey of Meiosis I. As you can see, it starts with one cell and ends with 2. And it all happens over 4 main stages, which have these fancy names: prophase, metaphase, anaphase, and telophase I. Let's break down what's actually going on in each of these steps. Okay, let's walk through this. In prophase I, the chromosome pairs find each other and literally swap pieces. Then in metaphase I, these new, shuffled pairs line up totally randomly in the middle of the cell. During anaphase I, the pairs get pulled apart to opposite sides. And finally, in telophase I, the first division is done and we're left with 2 brand new cells.
Now, let's zoom in on that really cool event in prophase I, crossing over. This isn't just a simple meet and greet. It's a physical trade of DNA segments. The chromosome you got from your mom literally trades sections with a matching chromosome from your dad. What this does is create completely new chromosomes that are a mosaic of both your parents' DNA.
This visual here shows it perfectly. See how the chromosomes from each parent, the blue and the pink, they get tangled up and swap segments? The result is what we call recombinant chromosomes. They're a unique patchwork of the originals. This right here is our first major source of genetic variation. It's incredible. But wait, there's more. That's not the only way variation gets cooked up. The second source is a concept called independent assortment. Remember how those pairs line up in the middle in metaphase I? Well, how they line up, which side the mom chromosome or dad chromosome is on, is totally random. It's a 50-50 coin flip for every single one of the 23 pairs.
So, what does that randomness actually translate to? Get ready for this. In humans, with our 23 pairs of chromosomes, this one step alone creates over 8 million possible combinations. And that's before you even add crossing over to the mix. The number is just astronomical. It's why every single sperm and egg cell is a completely unique genetic lottery ticket.
Okay, so after that great shuffle of Meiosis I, we're left with 2 cells. But the job isn't done yet. Now we move on to the second act, Meiosis II. And you can think of this part as a much simpler final splitting process. You know, meiosis II actually looks a lot like another process you might have heard of, mitosis. Both of the cells that we made in meiosis I now go through this second division. And the main goal here is really straightforward. Just separate the sister chromatids. Those are the identical copies of each chromosome that are stuck together.
And this brings us to something that trips a lot of people up. What's the difference between anaphase I and anaphase II? Well, the distinction is actually simple but super important. In anaphase I, entire homologous chromosomes, the pairs, are pulled apart. But in anaphase II, it's the individual sister chromatids that get separated. It's a totally different move.
So, after all that, what do we have? The grand finale of meiosis gives us 4 daughter cells. Each one is haploid with half the original chromosome count, and, most importantly, thanks to all that crossing over and independent assortment, every single one of those 4 cells is genetically unique, not just from the parent, but from each other.
Now, to really lock all this in, let's put meiosis right up against mitosis. They're both types of cell division, sure, but they have fundamentally different goals and totally different outcomes.
This chart lays it all out beautifully. Mitosis, think of it as a copy machine. Its purpose is growth and repair, so it makes 2 identical diploid cells. Meiosis, its purpose is sexual reproduction. It goes through 2 divisions to make 4 different haploid cells. It halves the chromosome number and uses crossing over to create all that awesome variation. The bottom line is simple. Mitosis clones, meiosis creates diversity.
And that really brings us right back to its core mission, doesn't it? Meiosis does 2 absolutely essential things. It creates the sex cells we need for reproduction, and it generates the genetic variation that fuels all of evolution. It's pretty amazing. So meiosis is this incredibly precise, elegant dance. But, like any complex choreography, sometimes a step is missed. What happens when the chromosomes don't separate correctly? This is a phenomenon known as non-disjunction, and the consequences can be profound, sometimes leading to genetic conditions like trisomy 21. It's a powerful reminder of just how critical this perfect cellular performance is for life as we know it.